So the point of, is this working? Sir, is this working?
Yes.
Pardon?
This is only for recording.
This is only for recording.
Yes. Because there are two mics. I think this is the one which you have.
Yeah, but then I have to just stand here to use this. But okay, that's fine.
If they are having some problem.
Yeah, okay. So what we talked about, this so-called enhancer, super enhancers. So this is as if that DNA is a naked DNA molecule onto which these proteins can bind to and do whatever they want. But in eukaryotes, I'm sure you have heard about what's called the chromatin. Yeah? So tell me something about it, since you all have heard of it. What is chromatin? Hold on, we'll start here. Yeah. Right. So in eukaryotes, so chromatin plays at least two roles. Number one, that so there is a histones, 4 of them. 5th is there to higher order structure. So the 4 of them will make a nucleosome. Nucleosomes are octamer of these 4 histones. Which are those? Yeah, don't say H1. H2A, H2B, H3, and H4, right? So 4 plus these 4 will make an octamer around which our friend DNA wraps around. This is one nucleosome, next nucleosome, and so on. And then H1 comes in between here. And the point of this is it leads to formation of high order structure or packaging of the DNA. Packaging is very important because imagine, I'm sure you have eaten long noodles or spaghetti sometimes in your life, right? So it's just if you have just very long, right, it's very hard to manage it. So inside the cell, the packaging is necessary to have all of that very long piece of DNA stuffed into the nucleus. Yeah? But now, after solving this problem, there is one more problem, and that is for either DNA replication or transcription, you need to have access to DNA, right? So now there's a problem. So chromatin plays an important role in gene regulation in addition to packaging the DNA. And so I will not discuss a lot about these because I'll assign you a paper and you are going to talk about something about the plants and the chromatin tomorrow. So the plan is that sometime during the 4 and 5 p.m., yeah, you will be assigned some article, yeah, and then you have to come up and give in about 5 to 7 minutes an executive summary of that so that everybody can benefit from combine of those articles. So anyway, so the point here is that chromatin plays an important role at different parts of the plant life cycle, yeah, in terms of activating or repressing genes. And that's an example which will show up in more detail is that indeed something critical happens that will lead to expression of the right sets of genes for defined parts of the plant. And in either of these, so whether you are in the apical tip of the shoot or the root, yeah, there are these special cells, yeah, called the stem cells. So who has heard of stem cells? Okay. Who hasn't heard of stem cells? So then tell me, what are the 2 properties of stem cell? So stem cells are undifferentiated, and the point is that they can give rise to, at least in human beings, yeah, in animals, all kind of other cell types, yeah. And a pluripotent stem cell is what we call is something that can give rise to all the 3 germ layers, ectoderm, mesoderm, and endoderm, and similar things applies to stem cell in plants, and there are lots of them. So a stem cell, number one, it can renew itself. So ability to regenerate itself is one key property of stem cell. Yeah, and that has to happen throughout the animal and plant life. And they also give rise to one daughter cell that we call it transient amplifying cell. And what they do is it regenerates, multiplies, and then they divide, and finally they differentiate. Yeah, and you keep on this reservoir of stem cells to take care of the needs of the plant, either normally or in response to some kind of a injury or change. And this is simply to say that it plays an important role in the human or cancer cells. So in what you are going to read, these players will look important because these are the chromatin remodelers, right? They play a role in both development and differentiation of right kind of plant parts. And hopefully you will talk about one of these in significant detail, about one example of the details as to how differentiation / organogenesis can happen, who does what and how. Yeah. And this point, this is simply to show that this word chromatin plays a real major role in regulating genes, and only then the right things happen during plant development and growth. And what you can see is you can have many arrows going either out of these or into these. So these are activating arrows. They turn or activate that particular target. Yeah? And these other signals, which are this line and one more line, so these are what inhibits that function. Yeah? And you can see there is a role of phosphorylation and dephosphorylation. So this hub does lots of these things to then eventually lead to activation of right sets of genes in the context of chromatin. And I should point out that, have you heard of this word switch sniff? Those who have heard, raise your hand. And you know who discovered this? So I was lucky to be working in a department where this guy named Craig Peterson, the one who discovered switch sniff, yeah, and he actually taught me yeast genetics back in 25, 30 years ago. So he worked in Ira Hershkowitz's lab at University of California, San Francisco, working with a simple experiment in yeast, and that led to discovery of this chromatin remodelers. Yeah. So until that time, people were studying transcription, but they were all studying transcription as if DNA is a naked molecule to which these activators and repressors can bind to at will. And what he told was, and then that led to another 10, 15 years of, that's what filled up this whole place. There are many players after the discovery of switch sniff, was that this is a very complicated place that's subject to both active or positive and negative regulation of these genes. And so our cells, just to give you an overview, it responds to both external and internal signals. Yeah? So the extracellular signals are such as you have something outside the cell, it sends a signal. So let's just say in case of the insulin signaling, so the insulin will bind to its receptor, that leads to phosphorylation, that leads to this whole cascades of RAS, RAF, MEC, and so on, and finally controlling activity of a transcription regulator. Remember we said activator or repressor? So now these are the final targets of anything that cell receives at the outside, and that signal is sensed, transduced across the membrane, amplified inside the cell, and finally leading to a long-term effect on gene expression. That applies to animals, that applies to plants. Same thing is chromatins, even though these chromatin remodelers are now also present in plants, but they were originally discovered in HeLa cells and before that in yeast cells. Yeah? So these are universally considered player in plant development as well. So I'll go through the next set of just an overview of transcription, not where gene regulation ends. Yeah, that's just one of the steps of gene control. And we'll come back to this at the end of on Friday, where you have to tell me what all you can do or what ideas you have where we can use computational, molecular, or whatever biology to interfere or do something about these steps. Good? This part I'll go kind of quickly because I was very slow deliberately to begin with, so we all are comfortable with each other. So as I said, transcription has been studied a lot. Yeah, it plays a key role, but that's not the only regulation, and that's where my research happened, and I'll talk about after lunch about what I have done or what things I have discovered in my life. So this is a slide that DNA makes RNA makes protein. Now you can see that before, this is what we talked about, how transcription can happen by polymerase, by activators, by repressors, by superenhancers, and so on. And now you can see that this is squished into a very tiny place. So now when the RNA gets made, there are lots of RNA binding proteins, and they play a key role in how genes are expressed and genes how they are regulated. Because as they say in life, the devil is in the details. Meaning that when you go and talk about what you want to do, you need to understand the in-depth of all of those things before you can do something good about the crop improvement. So an mRNA gets made. In eukaryotes, it has exons and introns, and you can see based on these shapes or the colors of these molecules, so these are the RNA binding proteins. So as soon as the RNA gets made, a bunch of proteins immediately start getting loaded onto that, and they play important and key role. But the point here is that what you will see is that some proteins come and bind, and they will remain all the way till the end with the RNA. There are other proteins, they bind early on, but then they leave somewhere halfway. And there are other proteins which will come back after, and then they leave. So the point of that is exactly it says that it is a very complicated process even after transcription before an RNA can be translated into the proteins. And lots of these RNA binding proteins play a key role in regulating that gene expression and each of these steps, splicing, polyadenylation, RNA export, and transport into the cytoplasm, and finally translation. Each of these steps can be regulated. Yeah? And so in your, again, crop improvement, yeah, each of these players can become important role depending on what your goal is. So I'll just touch upon literally this one guy, but the point of that is RNA can be controlled at many of these steps, including capping, polyadenylation, modification, editing, export, stability, localization, and translation, and so forth. So I'm sure you've heard of introns and splicing, yeah? But the question, do you have a question? Yeah. But the question is, how do you think introns were discovered? Pardon? How about now? What's that? Right? So, and that led to the figure I'm going to show you. This led to one Nobel Prize. The question was, why was it worthy of a Nobel Prize? It was because until this time, which is 1977, yeah, we knew that genetic information is collinear. Collinear meaning that once you know the sequence of DNA, you can figure out the sequence of RNA, and once you know the sequence of RNA, you can figure out the sequence of amino acids in the protein. Yeah, because once you know the start and the end, in between it's all predictable. But what these guys did was they discovered, and I won't talk about this experiment, but this figure is the one that gave Nobel Prize to Phil Sharp along with Richard Roberts. And Susan Burgett was a professor at this place called Baylor College of Medicine, and you'll hear a little bit more about that after the lunch break. Yeah. So she in fact was the one who taught me splicing, and it was her work. She is the first author on this paper that gave the Nobel Prize to Phil Sharp. So the point here was that they discovered there is a sequence in the DNA that gets into the RNA but never shows up in the protein. So it is no longer collinear. And again, we have to revise our molecular biology and biochemistry textbooks based on that discovery. So introns, exons, they are spliced together to form this mature RNA, and it is because there are these what is called splicing signals, 5' splice site, 3' splice site, and so on. These signals are recognized by a bunch of factors, and that leads to splicing. So now get ready for a question. Imagine you have this is exon, and this is intron in between, yeah, and this is going to be removed, yeah, and exons joined. Now the question is, you have a substrate, exon 1, exon 2, and 1 intron in between, yeah, and suppose you have all the goodies in a test tube. You take your RNA, put in the goodies that can splice the intron out, yeah. So you give it a precursor, you splice it, and now the question is, your RNA radio labeled, what you are going to find? Don't look at there, yeah. So what you are going to find when you do the splicing in a test tube? Two. So we have this, right? Yeah. Now tell me, I'll exactly do what you want me to say.
I had a big strand.
Exon, intron, and exon, all will be spliced out.
The middle intron is spliced.
So this is out, and these are joined. Yeah. So we are starting material as this one. It got converted into this plus this. So how many things we got? Starting material and 2 products, so total 3. Yeah. So now, when they did the splicing and ran a gel, we were expecting 3 bands. Good so far? And what they found was this. So they had a band here, and I think we can call this our starting stuff. And they found a band over here, a band over here. Ignore this, is just the background from RNA degradation, not important for the experiment. But how many bands you have here? Total in one lane. So 1, 2, 3, and 4, and 5. So now we have a problem. So in America we call it Houston, we have a problem. You know why that's called Houston, we have a problem? Huh? Because Houston is what's important for where the NASA is when they send stuff up there, right? So now, remember Apollo 13? Yeah. So the call was, when things went bad, they said, Houston, we have a problem. Yeah, and it was bad. So, my friends, we have a problem here. That we agreed on 3, the way you asked me to do, right? But now, when I did the actual experiment, we got 5. So now the question is, suppose you are, as a part of your PhD, did this experiment, yeah? And now what does it mean to you in terms of how introns are spliced out? Because this is what led to that Nobel Prize discovery. Hold on, I'll give you a chance. We'll start from our friends over here. Oops, I don't know what I did. Okay.
So there might be alternate splicing.
So there is no alternate splicing because there is only 1 intron here. Yeah, 1 intron. It can be either spliced or not spliced. But I'll come to your alternate splicing. Your wishes will come true, but in just few seconds. Anything else? Yeah.
The exons remain
Axons remain separated. All right, so what he's saying is that we had this starting material, we got into 1 plus 2, right, the second one, but what he's saying is instead of this, what he wants us to do this. So now we have starting material, number 2, number 3, and number 4. That's what you want, right? Okay, I still need one more. Anybody from this? Well, but so we have 1, we have 2nd, we have 3rd and 4th, and we have 5. Okay. So at least we have counted correctly, right? Number 5. 1, 2, 3, 4, and 5. But the problem is that's not correct.
So, sir, intermediate of splicing.
Perfect. So the key here is that if you look carefully at this experiment, yeah, imagine this is your PhD experiment. What you should be asking is, you literally scratching your head that day morning, because there is something here that does not make any sense. Yeah, what that thing is, this is our pre-mRNA. Yeah, now we can find these 2 small ones, maybe these separate ones and the 2 together. Yeah, so they will both be smaller than our starting stuff, right? Because they are pieces of this work stuff. But the problem is, in this gel, there is this band and this band. In fact, we have 2 bands, even though we are happy with the number 5, but 2 bands are bigger than our starting stuff. Pardon?
So ligation of exons, extra exons.
Right, but I need. Let's just see if this helps. So we are good. Exon, exon, and let's just call it, just to make it easier, this is 100 nucleotides, 50, and other 100. Let's just make it one of them 200. Yeah, so we can separate the sizes. So this is how long? 200, 100 is 300 plus 350. Yeah. So now, if we keep, this will be 50. So migrating slow, faster. This is 200. It will be migrating slow, faster, right? Lower in the gel. This is 100, also lower. If they are together, what you are saying. So now it is 200 plus 100, 300. That's 50 less than this, right? So they all will be smaller, no matter which way you do it. Yeah. So the problem was that if that these are easy to account for, yeah, and in fact, if suppose these bands showed up here and here, yeah, somewhere here, no problem, because on that morning when you saw this gel, you said that, oh my God, I got my 2 other things, this alone out and these 2 together. Who cares for the other things, right? But then you have missed the important thing. So what happened here is that these bands are migrating weird, yeah, unexpectedly or surprisingly. So these are not bigger than this starting material, but they are migrating differently because they have a weird structure, and that's what we call is lariat intermediate. And what that intermediate is, that it kind of forms a, so suppose this is my starting stuff, right? And now imagine this was my exon 1. So just think it is still there somewhere in the air, but now this happens as a part of splicing. And remember, if these bands never showed up at the top, they would have never figured out lariat. But because then they would have said that, oh, these are some kind of degradation products, yeah, and who cares, yeah. But because they were above, so they couldn't have been degradation products, right? They are something important, yeah. But what are they? They are, so one of them is this guy. Now, if I have the second part over here, at some point this exon also will go. So starting, 1 is gone, but this is formed, then second is gone, after it is joined to this guy, and now you are left with just the intron in this structure lariat intermediate. So these 2 are lariat intermediate, intron connected to this exon 2, and just intron alone in this shape. So because of this loop structure, they were migrating differently, and they showed up over here. Because again, if you came that day, you went and talked to your advisor, he will say, what wrong you have done? Nothing is supposed to be bigger than the starting material. So that is because of, anyway, so this is what it is. That's now we know how the splicing happens. And splicing, just like transcription, it involves lots of proteins and lots of goodies to do that. And this is just a very complicated way to tell a very simple thing, that of course we have exon 1 intron and exon 2. Now U1 SNRP comes to bind to this signal, then next guy comes to bind to this signal. You form what we call as the commitment complex. So that means now this intron is committed to splicing. It cannot go back. And after that, the whole spliceosome, which is a very big molecule here. In yeast, it's about 60S and 40S in yeast. And you know how big are ribosomes? Right? 60, 40, they become 80, yeah, and so on. So anyway, it's a very large complex. In that large complex is where, so now you see this one arrow, and this is out. So so this one arrow, that leads to first step of splicing, to form lariat intermediate. The second step is where the two exons are joined together. The two exons are joined together, intron is released, it gets debranched and degraded. These exons are joined together, RNA goes to the cytoplasm and get translated. This is a quiz question for you, that suppose one night you had a dream. The question was how those splicing signals are recognized. Yeah, we call a 5' splice site, 3' splice site, and something else. Yeah, so 5' splice site recognized by U1 snRNA. Yeah, now the question is how is it get recognized. So what you found was that there is a sequence in U1 snRNA that is complementary to 5' splice site. Yeah, so you had this dream, you don't know this experiment, right? And now you come to the lab next morning and you want to experimentally show it. And you had the dream that U1 snRNA recognize the 5' splice site or a splicing signal by RNA base pairing, shown over here. Because what you noticed was there's a sequence in U1 that is perfectly complementary to the 5' splice site. Can you see it here? Yeah? This is in your dream, and now you have to experimentally show that this RNA base pairing is indeed important for splicing. So the question is, what next experiment will you do? You'll get a nice publication in Cell or Nature. You get a PhD. Life is good. You move on with life. What experiment will you do? And all of these are to excite you that you find a problem, and then what experiment will you do to experimentally prove it or show it? What will you do?
What sequence is complementary to 3 days? This is for 5 days, and what sequence is complementary for 3 days sequence?
So hold on. So this is a sequence of our mRNA. This is where the intron starts. So this is a 5' end of the intron, and what you see is there is something in the U1 that is A paired with, or A can pair with U, C can pair with G, and so on. So now the question is, how do you know this actually happens in the cell and is part of the splicing reaction? Pardon?
Hybridation. Can we prepare? You can prepare for hybrid like normal body.
So if you prepare the probe, right, and you hybridize, maximum is going, if you see the signal for hybridization, the only thing it's going to tell you is that yes, this complementary sequence can hybridize, but it doesn't show that it does hybridize, right? It can hybridize, and there's a big difference scientifically, right? Can hybridize versus it does hybridize. So how will you show it does hybridize? And that's important for splicing. If you are working on splicing in the 1980s, you think that's an interesting question, because you just do not know how does the cell finds these introns and removes them out. This is the way to do that. So what they did was that you took this sequence which you suspect can hybridize, right? So now you make a mutation of, you change this A to a U. Now A was paired with a U, yeah? Now you change this A in the intron into a U, yeah? What you are trying to do is, you are trying it to not, because U cannot base pair with a U. Remember our Chargaff rule? A pairs with T or a U, G with a C, life is good. So now you are trying to break that complementarity. And now when you do this mutant, yeah, you test it. You can have one of the two possible outcomes. One of them is splicing is not affected, yeah? And that will mean to you, what will you conclude? That the dream that you had, it was just a dream, right? But the other outcome could be that when you do that, the splicing is dead. It no longer splices. So then, can you conclude that indeed this sequence base pair with this? Those who think we can conclude that by making this mutation, splicing is dead, we can conclude that UN base pairs with 5' splice it. Raise your hand if you can conclude that. Raise your hand.
It is not final conclusion.
You don't agree with that?
No, so it cannot be the final conclusion.
Because?
Because it means some point mutation can also cause.
Perfect, right? So the point here is that all you have shown by this experiment is that changing that A to a U is bad for splicing. So it's a big deal, right? So you have not gone far enough in your experiment or the way you designed it to show. So what you need is one more step, and that step is that suppose now this splicing is dead, but now you go and make a mutation of your U1 snRNA sequence also and change that from U to an A. So now you have restored base pairing. You change A to a U, but now you change this U to an A. Now again two possibilities, right? One is it does not restore, then you cannot say that it was an old base pairing, right? It could be it's irrelevant, right? But suppose what you find from this experiment is that when you change this to U to an A, now the splicing is back. It happens. So what we call is that you have made a compensatory, mutation, and if that restores splicing, then you know that this is important, the base pairing happens, and that's exactly what happens. And that was a Cell paper published in early 1980s. It showed that because we knew there are introns, we knew there are exons, and we knew exons are joined together, but now no one knew how those are recognized. People found those splicing signals, and with this experiment, this was the key first experiment to show that indeed U1 SNRP or U1 snRNA recognize the 5' splice site, and that is our friend here. So this is what I just showed experimentally. So to put this over here was based on this experiment, and they did the same experiment where they showed that U2 binds to this sequence called the branch point over here, and then the whole spliceosome assembles, and then the two steps of catalysis happens, that the exons are joined together and so on. This I'm going to blow my horns here. So to show that U2 recognizes this sequence called the branch point in the intron, that also requires base pairing. So what we published a science paper many years ago where we showed that a splicing factor, its name is U2F, that you don't need to worry about now. After lunch we'll talk about that. So what we showed was that U2AF does something to recruit U2 snRNA to this branch point sequence, and then only splicing can happen. So we talked about our friend Tom Cech, the guy who discovered ribozymes, and he got Nobel Prize for that. And it was fortunate that he's the one who hired me at University of Colorado Boulder. So this was the experiment that led to a Nobel Prize for University of Colorado and to Tom Cech, and the discovery of ribozymes. So what they did was, so there is a tetrahymena, group 1 intron, and you have this substrate over here, and what they did was they took that in a test tube and let the splicing happen. Now what I just talked about over here, there are these 5 SNRPs, U1, U2, U4, U5 and U6, plus hundreds of more proteins which together form that 60S complex. Yeah? What he showed was that all you need for the splicing of this intron is simply your substrate. Yeah? None of the proteins that I just talked about. If you simply add GTP to the reaction, without GTP, no splicing, just simply GTP, it allows splicing. So anyway, and all it can happen in a test tube. Your intron plus this, it is spliced out. So that's called self-splicing of group 1 intron. And what it showed was that intron is catalytic. It can do the chemistry of removing itself. So the discovery of ribozyme that solved our so-called chicken and egg problem we talked about before, right? Because now we know RNA can do things like proteins, yeah? And RNA can also be genetic information. And that's more details of that. Group 2 also introns are there. So group nuclear pre-MR introns are where you have this and this. These are 2 steps of splicing. Tom checks group 1 is this, where the GTP from outside provides a hydroxyl group to do the first step of splicing. That releases this 3' hydroxyl over here to attack the end of the intron to join the 2 exons. Self-splicing, no proteins needed. But now this group 2 are kind of in between, that they look like this mRNA type of introns, that they have this branch point within that. They don't need the GTP from outside, but they are like this one, that they also don't need any proteins of what these guys need. Yeah? So discovery of RNAs which can do chemistry of different kinds. Now we'll come to your question of alternative splicing. So as we know, splicing is important because if you can't remove introns, you can't make proteins. Very simple. So when the genomes were sequenced, and we'll talk about that after lunch a little bit. So what we found was that of course introns are abundant. Most of our genes have introns. In fact, most of the genes in most of these other model systems have introns. You can see even in the plants there are plenty of introns here. Now the problem here is that if you look at approximately the number of genes.
Lunch break will be at 2 o'clock because the food will be ready by 2 o'clock, so you can continue up to 1:45 or something like that. Okay. Is possible.
Not at 1 o'clock.
Sir?
Yeah, that's fine.
Actually, another training is going on, so they are arranging.
That's fine.
Sorry, sir.
No, it's okay. So I have been told, whispered in my ears, that the food that they ordered is not here. So that means I was trying to make it fast so you can go to your food. He's saying, slow down. You don't have a plane to catch. Yeah? All right, I'm going to entertain you for the next hour. Okay, where I was. So when the human genome was sequenced, what they found was, so they were expecting some 150 to 200,000 genes. Yeah? When they actually sequenced the human genome, guess what they found? 25,000 or so genes. So there were much less number of genes than they were expecting. Yeah? So now we have a problem, Houston, again. The problem is human beings are quite complicated and complex, right? So if you look at the fruit flies, they have about 13,000 genes. Nematode worms, yeah, they have about 19,000 genes. And we, who we think are much smarter than these tiny insemincy guys, right? But we only got these many genes. So now the problem was, how do we become more complicated with roughly the same number of genes? Yeah? So that's where alternate splicing plays some important role. So alternate splicing is abundant. In fact, most of our genes undergo alternate splicing. And that's what I studied for the last X number of years. And after lunch, or depending on when the lunch comes in, I'll entertain you with all of that stuff. So what is alternate splicing? Anybody here?
Sir, some of
Okay. So we start out with an mRNA which has exon and intron. Yeah. So what we said is that that intron has to be removed. Only then you can make protein, right? So now we are saying, so I said, okay, the cell has figured a way out that it knows how to find those splice sites and remove the intron and join the exons. Yeah. So now it can make protein. But now what we know is that not all introns are removed all the time. Not all exons are included or joined all the time. Not all splice sites are also used all the times, which means in one word, life is pretty complicated. Yeah? In fact, if you take a piece of paper, yeah, and start drawing all the combinations, anything that you can think of exists in the cells. Just to give you a feel for these. So normally, this is what should happen. You should join this exon to this exon and this exon to this exon, right? Where you have this plus this as a mature mRNA. But we have examples of where it's a skipped exon, that sometimes you have this way, but other times you go from here straight to here, skipping this yellow, green, whatever color you call it, part. So that's alternate splicing. Yeah. So you are using this exon only some of the time, only in this way of splicing. But when you use this way, you skip it. There are examples where you can have this splice site joined to over here, this splice site joined to over here. But other forms are where you join this over this and this over this. So these are mutually exclusive exons, meaning that only one of these two can be in the RNA, but not both. In other words, as the Raja of jungle says, there can be only one sher in the jungle, right? So these are exactly the same thing. They're saying only one of can be there, but never both in the same place. We have also examples where the same 3' splice site can be joined to either this or to this splice site, 5' splice site, or where a 5' splice can join to either this 3' or this 3' splice site. Or we have examples where this intron can be sometimes removed, but other times not spliced out. And again, in those genes, that's very important for that gene function and gene regulation. Yeah. So anyway, all of these things that you can at least write on a piece of paper, they are there in the cells. And now the question is, so how does the cell know what to do? Yeah. And that's our splicing regulation, just like our transcription regulation. So some concepts are exactly the same, some are unique to splicing. So these are where you can make either this mRNA or that mRNA from the same gene, yeah, final mRNA, by alternative splicing. But we have examples where, if you look at this gene called slowpoke, you can make more than 500 different mRNA types by alternative splicing. And that's a lot, yeah. But that's not the best. If you take the neurexin gene that works in our brain, it is involved in making the synaptic connection between the neurons. What you can do is, from the same gene, you can make over 1,000 different type of alternative mRNA types, and therefore protein types. And this is what I call as the mother of alternative splicing, DSCAM. So this is a gene in Drosophila. From one gene, at least in theory, you can make 38,016 mRNA types. And that number is pretty impressive. Let me give the context. In Drosophila, there are about 13,000 predicted genes, yeah, mRNA genes. This one gene itself can make 3 times as many mRNA types than the total number of predicted genes in Drosophila. So it's a big deal. I wasn't planning on doing that, but because there is no lunch here, so the question here is, based on, so you found that this is our gene called DSCAM, yeah? And you had a dream the night before, and this was published, your dream was published in Cell paper down the road, yeah? So the dream was that you can make 38,000 mRNA types from this gene. Next day you come to the lab or to the bench, and you have to experimentally show if these are indeed made. So the question is, what experiment will you do to find out how many of these, what we call is theoretical mRNA?
So can we study the proteins? How many proteins are produced?
How will you study the proteins?
mRNA can be compared with the original gene.
All the series are exactly the same. This antibody, or let me put it in this way. This red stuff is what is called transcription in the plasma membrane. Practicals? Okay. So Drosophila is a good model system. The reason we study Drosophila is 80% of human genes which are linked to some human disease are found and studied in fruit flies. Yeah? In fact, for most of the human important genes have been discovered and studied in fruit fly before they were known to be important in human systems. So in Drosophila, alternative splicing is what controls sex, how they become males and females. Yeah? So this is more complicated than I would like to, but the point here is that you can see these two ways, a pathway of this type or this type. So focus on this part. So how much, and this is a right connections because of this. So I told you about alternate splicing this way in male. In females or XX. So we talked about splicing, alternate splicing and its importance. There's also in nematodes and trypanosomes what is called trans-splicing. Anybody heard of trans-splicing? Anybody who has never heard of trans-splicing? So you all heard of it. All right, so what's trans-splicing? Our friends over here. That's okay. Yeah. So if I ask you this question, how was trans-splicing discovered? Yeah, I think some of you are very inclined to say we'll do transcriptomics, right? Imagine life before next generation sequencing. Yeah, you are a PhD student, you are about to discover trans-splicing, you'll get a nice cell paper. Yeah, question is, what experiment you might have done before next generation sequencing that will tell you that trans-splicing exists. So the idea here is to give you a flavor for all of the steps in gene regulation that can be targets for future crop improvement or future agricultural experiments. So how was trans-splicing in nematodes discovered? So if you suppose take mRNAs from these cells, yeah, and you sequence them, you make series sequence them, yeah. You sequence the first one, you publish that nice sequence, yeah. You sequence the second one, you will publish that also, but you will fail to notice one thing: the 5' end of mRNA is exactly the same. So that wasn't obvious in the second mRNA sequencing. But when people sequence lots of mRNAs, and then some smart guy, or maybe some guy like me who has nothing else to do in life, yeah, he was just staring at these sequences, and he said that, you know what? There is something funny going on here. That you have this gene sequence, right? Gene A, gene B, gene C, and they of course are all different, but the 5' end of these are exactly the same sequence. Now, when they sequenced the genomic DNA, what they then found was that extra piece that's there, it is actually not in the gene. So meaning it has just come out of thin air. So the thin air was that suppose you have gene A, and what they find is that after it is splicing, you only see this part. And when you sequence this, you find this plus attached to these few nucleotides over here. And you sequence its DNA, and what you align the cDNA and the DNA, and what you found was that the alignment was perfect from here all the way until where the yellow thing ends. And now there was no sequence called blue in the genomic DNA. And what they found was that your mRNA2, cDNA2 also has, of course, a different sequence that matched perfectly to its DNA, but there was something extra. And finally someone picked up that, this extra sequence is common to all of these mRNAs. And that led to there has to be trans-splicing. And what was it? That there is a gene that makes this 5' half of the intron and the first exon. Yeah, that gets made, and that by trans-splicing gets added to every single, or at least those RNA which are trans-spliced in the cell. And the RNA is capped with a 5' cap, and I'll talk about this later on. So we'll end at least this part with this experiment. So you study splicing, and you're trying to understand some things, and I'll tell you what that thing is. So what you do is you make an RNA using this promoter over here. So you make this RNA, and when you are making this RNA test tube, you use this kind of a cap structure, M7GPPPG, because this polymerase called T7 or SP6 can use that cap analog to make this RNA with this cap, exon, intron, and so on. Yeah. Now you use a different test tube, and you put a GPPPG cap analog, and you transcribe this RNA, and finally an RNA that doesn't have a cap, so this RNA. Then what you do is you have this in vitro splicing that you can do in a test tube. So you take each of these, so this goes here, this goes here, and this goes over here. You splice them and run a gel. Finally, when you run gel, you find this is our pre-mRNA, yeah, and these are other splicing products. And now the question is, based on this experiment and this data, what can you conclude? Can be degraded. But here, based on what you think the mRNA, so that means that this RNA has to be degraded. Yeah? But do you see evidence of degradation here? Because, so this is our precursor. So do you see any difference in the RNA stability here? Because you see that precursor everywhere, right? So even though that conclusion is correct, that cap is important for RNA stability, but this experiment doesn't tell you that. So this one, this is splice, huh? So splicing and here, no splicing. Yeah. So will you conclude that cap is important for splicing? No? What will you conclude? Pardon?
Capping is important for splicing.
Is important.
Capped one is getting degraded, that's why splicing is not occurring.
Well, it's not degraded, it is still there, right? So if you compare this versus this, what you see is that in this cap and this cap, you have spliced RNA, right, where 3 exons, in this case, exon 13 and 14 are joined, but this intron is not removed. Here, exon 13 is not joined to 14, but 14 and 15 are joined together, right? Here, 13, 14, 15 are all joined together, both the introns are removed. So what you will notice here is that the cap, whether it is M7G or just the G, right, capping still happens. But when it is not capped, then what you see is that there is very little compared to this signal over here. There is very little, if any, of this product. Yeah, but this product is there just like these two. So if cap was important for splicing, then you should not have even gotten this band over here. But we do get one of the splicing products. Yeah. So the conclusion here is that the cap is important, but only for the splicing of the first intron. Cap has no influence on the splicing of any other intron downstream. Yeah? And let's go to the other end of the RNA where it is poly-A-tailed. So remember, all the mRNAs are poly-A-tailed. All we agree on that part? So, pardon?
Histone mRNA are not.
Yeah, except for the exception of the histone mRNA, every other RNA is poly-A-tailed. So the experiment here is that for polyadenylation, we need a signal called over here AAU, AAA. Yeah, so we all good on that? So the experiment here is that this is our wild type with this intron, this exon, intron, exon, and a wild type poly signal. Yeah, we have one more here where what you have done is you have changed the signal over here from AAU to AAG. Yeah, and finally we have just put in some unrelated sequence. Yeah, so we have three substrates, three RNAs, and now you splice and polyadenylate, and what you see over here is this. So let me explain. So this is our starting stuff. Yeah, this is our wild type, this is our mutant, and this is where the sequence is replaced by something unrelated. So the question is, what do you conclude from here? And because there are just too many bands over here, so I'll let you digest that. What you'll conclude from here is that you need a good wild type signal for splicing to happen. That is strange. It is surprising because splicing is something independent, right? Or that's what we used to think. Polyadenylation is an unrelated RNA processing event. But what they found here was that you have your two exons, intron in between, now poly-A-tail over here. So it should be spliced regardless of this. And this will be poly-A-tail regardless of what happens over here. But what they found was that if you look at, and again, go and I'll post these slides for you. The point here is that you see bunch of bands over here in the wild type, and some of them are either spliced or polyadenylated or both. That's why these multiple bands. So what you can see here is that when you change that AAU, AAA to AAGAA, now you can see there is no polyadenylation. That's expected because you have just messed up polyadenylation, right? But surprising thing was that you mutated poly signal, which is way downstream. It affected the splicing of the intron, which is upstream. So that is surprising. So that means that splicing and polyadenylation, they talk to each other. Yeah. Now that's not shown over here, but suppose you do one experiment the way we did for the capping. The experiment is here it's only one intron, yeah, but suppose there are two introns, exon, intron, exon, intron, and our poly-A-tail here. There we tested the role of cap or no cap, and we said cap is important for the splicing, but only of the first intron. If you do the same thing now for this, right, to have, and that experiment is done, I just haven't shown it here, that you mess up this poly signal here, what do you expect to find? In this case, what we said was if you mutate that, no splicing. I'm saying is, suppose you do this experiment next day, what you're going to find? So what was found was that when you had the wild type sequence, this was joined and this was joined. Life was good. It was poly-A-tailed, just like in that wild type sequence, right? But when we mutated it, then what happened was this was joined, but this was not joined. So that means poly-A signal or the poly-A machinery is important, but for the splicing of the last intron. Yeah? And that brings us to this guy, exon definition. So I want you to tell me on Friday what I just talked about here is relevant to what happens to the internal introns, right? I talked about the first intron, the last introns, but how the same logic also applies to internal introns. So that's your homework for Friday. Good? Let me check with our friends in the back about your lunch part, because they have told me to keep dancing here until the lunch comes up. Yeah, otherwise no lunch for me. What happened? The food is here or not yet? 15 more minutes. Okay. All right. So, so before we leave, so I hope